Here is a short lecture on an introduction to ecology. Ecology is the study of all components within a defined area. We're talking about both abiotic and biotic factors. Abiotic factors are things that are not living, and biotic factors are things that are living. Be careful not to confuse both abiotic with dead. Abiotic things are things that are never alive, like water, air, sunlight, soil. But biotic things can be things that are have been dead for a short time. So be careful with that. Ecological basic organization is the levels of organization that you learned about last semester. So biosphere is the largest. Okay, that's the entire Earth. And then a biome is an area that has a defined climate, an area with a specific temperature average, rainfall, and soil quality. Then an ecosystem is an area that has all of the abiotic and biotic factors in a defined area. A community is all of the living things in an area that is including multiple populations. And a population is just a single species. So all of the zebra that live in an area, or all of the lions in an area. Then a community would be all the zebras and the lions and the plants and everything else that's alive. Ecosystem is all of those living things plus the abiotic factors like sun and soil. A biome is all of those things that are in ecosystems in a defined area with a temperature average and precipitation average. And then again, the biosphere is the whole Earth. Individual is the smallest ecological level, and biosphere is the largest ecological level. So if you take a look at these again, this would be an individual, and then a community of groundhogs here. And this is, a population of groundhogs here. A community would be all of the living things in this area here. This kind of incorporates both ecosystem and biome. So ecosystem includes the fire and the soil and the air. But it's also a defined area. This biome would be a grassland. And then a biosphere is the whole Earth. So once we have that set system, we also see how that changes, and we can go through succession. Primary succession is going from nothing, no soil or anything, to a pioneering community to a climax community. So a pioneering community is the first organisms that live there, whether we have to wait for some erosion so that there's soil, and then little plants. It can also be a lot of lichens and mosses can be in that area. A climax community is the maximum mature community that it can reach. This is a complete ecosystem. What's a possible reason why there would be nothing there? Try and think of something for a moment. Well, the most typical reasons are melting glaciers, volcanic eruptions, a landslide, or a strip mine that leaves no soil at all, just bare rock left behind. So that happens if we have one of these options here, but sometimes we also get things that cause a disturbance, and then we have to re-establish the community. That is called secondary succession. So establishing an ecosystem after some kind of disturbance. What kind of disturbance do you think we would be talking about? Well, the most common ones would be flood or fire, where we still have soil left, maybe even some things remained alive. And so this happens much more quickly than primary succession does. We also can estimate the numbers of individuals living in an area. We want to know how big a population is for a variety of reasons. Maybe we want to know what the carrying capacity is. Maybe we want to help endangered species. Maybe we're looking at if we should allow hunting licenses, or how many hunting licenses we should allow. So it's a good idea to know how big our populations are, but it's not really feasible to go out and count every single individual in a community. So we're going to talk about 3 ways in which we can estimate population size. The first one is random sampling, and random sampling is when you take an area and you mark off a grid, and then you randomly select areas and then count the number of individuals in those areas. You add up those, find an average, and then multiply it by the number of boxes that you have. That will give you an estimated population size. But sometimes we get off by a little bit. So that can cause a problem if we are working with like clumped populations. Second way is point-intercept, where you take a line and you draw it out a certain distance and count the number of individuals that are on that line or a certain distance away from that line, and then multiply it out for the whole area that you are facing again. The last one is capture, mark, and recapture, and this is also called the Lincoln-Peterson Index. With this one, you have to use math. But what you do is you capture a number of organisms and you mark them in some way. Usually we use tags or spray paint, something that's non-invasive to those organisms so it doesn't hurt them. And then you release them back. Over a set period of time, you capture organisms again and see how many of them have been recaptured with their mark. This allows us to use a mathematical equation to determine how big the population is. This is very effective for organisms that are moving around, because if you try and use some of these methods like random sampling or point intercept, you can see that these guys are going to be moving around between quadrants, so you might have a little bit of trouble making sure that you're not counting the same one over and over. Whereas with this method, it's okay if they move. That's kind of the point. So that's all for this lecture. Please go ahead and work on your assignments and contact your teacher if you have any questions. Thank you.